Okay, should we get started, William?
Absolutely. But before we get started, yes, as we get started, I think the honor falls to me to welcome all attendees. And I would like to thank all the speakers for joining today. I'd like to thank the organizers, Caitlin Simpson from the Biophysical Society, for organizing this networking event. I think it's just a wonderful idea to bring people together with a common theme or topic, and for all the communicating with all of you. Noriko Shimizu here at NYU has done a wonderful job of setting up this online meeting, helping to advertise it, and she's been just a tremendous help. Now, the theme of the meeting, as you have seen, is really about new findings in ion channel biology, particularly as it relates to membrane proteins not previously appreciated to have ion channel function or channel function, and hence the name that we came up with. Now, before we get started, and I think this is in the next slide, there will be just a few comments about the logistics. Noriko, if you can go to the next slide, please. So there'll be no breaks during this entire symposium, and we're going to try to stick to the schedule that we've posted on the previous slide. There's going to be 4 talks. Stefan will moderate 2, and I will moderate 2. Speakers are going to have 20 minutes for their presentations. Now, if there's a loud gong going off at the, at some point, that's just a reminder for the speakers that their time is up. So don't get afraid. There's going to be 8 minutes to set aside for discussion. And we urge everybody to please use the question and answers icon at the bottom of the Zoom window. The moderator will read the questions. Now, there's going to be limited time, and we're not going to be able to get to all of the questions. So please keep your questions short and your comments short if possible. And if we run out of time and we cannot address all of the questions, then please feel free to privately contact the speaker either with chat or with questions and answers. And we hope that we can get to your question. Since we're short of time, I'm going to move on, and we're going to, Noriko, if you can go to the next slide, we're going to really introduce speaker 1 now. Now, there's been some controversies about the mitochondrial KTP channel in the literature, particularly as it relates to its molecular composition. And the recently published paper in the journal Nature by Diego De Stefani from the University of Padova in Italy has really shown how CCDC51 and the mitochondrial protein from the ABC superfamily really is the molecular composition of the KTP channel. We're looking forward to hearing what you have to tell us, Diego.
Okay, I'm going to share my screen. I hope you can see. So first of all, thanks, really big thanks to the organizers for inviting me to this very interesting event. So as William told you, I will talk about the molecular composition of the mitochondrial KTP channel. So usually when you think about channels, you always think about plasma membrane channels, but as a matter of fact, the intracellular membranes are crowded with channels, and they are as important as the plasma membrane channel. In particular, the inner mitochondrial membrane has a number of ion channels and transporters that are critical for their function. And this could sound trivial, but if you look to the original formulation of the chemiosmotic theory, you can see that the 4th postulate state that in general, the inner mitochondrial membrane should have a low permeability to protons and to ions in general. This, of course, this doesn't mean that that chemiosmotic theory is not true. As a matter of fact, the chemiosmotic theory is, of course, confirmed, and this only means that the ion channels in the inner mitochondrial membrane must be very carefully gated, and most of the time they should be closed and controlled very carefully. And the point is that there is the electron transport chain that generates a huge electrochemical gradient across the inner mitochondrial membrane. It's up to 200 millivolt negative in size. These represent a huge threat, especially for cations, because there is a huge driving force driving cations inside the matrix. And one should think that cation channels should be limited in the inner mitochondrial membrane, but as a matter of fact, there is a number of, if you want, on some way, unexpected cation channels in the inner mitochondrial membrane. There are channels specific for calcium, for protons, for potassium, for magnesium, and for any ions you can think of. And the point is that, of course, any influx of any cation should be counterbalanced by the efflux, and usually the efflux is mediated by a proton cation exchanger that exploit the proton driving force generated by the electron transport chain. In any case, my talk will be focused on potassium channels. And the case for potassium is that there are a lot of potassium channels that has been described to be present in the inner mitochondrial membrane. There are at least 7 different mitochondrial potassium channels that has been measured, for example, by patch clamp. But my talk will be especially focused on the MITOK ATP. So this channel was demonstrated to exist almost 30 years ago. There is this first publication by a Japanese group that performed patch clamp on isolated mitoplasts. And the year after, this paper almost demonstrated that there is this potassium sensitive channel that is inhibited by ATP, and the inhibition by ATP is somewhat reversible. And at normal ATP concentration around in the millimolar range, this channel is essentially closed. And importantly, this channel is also can be pharmacologically inhibited by sulfonylureas, so thus resembling, at least at pharmacological level, all the features of the plasma membrane KATP channel. And 1 year after, another group, the group of Keith Garlid, performed a similar experiment by using a partial purification technique. They were able to purify a potassium sensitive channels, ATP sensitive potassium channel, starting from mitochondrial preparation, and basically they confirmed all the findings of the previous paper. So they were able to purify these channels and to show that this channel is closed by physiological ATP concentration. However, in the next decades, there is somewhat interest around this channel. And the work of some groups, especially from the groups of Keith Garlid, Avan Sedgwick, and Brian O'Rourke, demonstrated that this channel is important in regulated mitochondrial function. For example, we know that mitochondrial potassium handling can regulate mitochondrial volume. By this, the entry and the homeostasis, potassium homeostasis across the inner mitochondrial membrane can regulate ROS production and ATP respiration. At the, let's say, organ level, what has been demonstrated by this group is that a pharmacological opener of the MITOK ATP is able to protect the heart and other organs from the so-called ischemia reperfusion injury. And most importantly, also pharmacological inhibition of this channel can protect can inhibit cardioprotection that is triggered by the so-called ischemic preconditioning. And we'll say something about this at the end of my talk. However, all these works were basically demonstrated using pharmacological tools. We now know that there are some drugs that preferentially act on the MITOK ATP as opposed to the plasma membrane KATP. As an example, diazoxide is generally used as opener for the MITOK ATP, although diazoxide can act also on the plasma membrane one. And 5-HT is the best described and, let's say, most specific blocker for the mitochondrial KATP. However, the point is that no one has ever succeed in identifying the molecular nature of this MITOK ATP. And as mentioned during the introduction, last year I found a protein complex located in the inner mitochondrial membrane that can account for this ATP sensitive channel activity. So this channel is composed by two different subunits, thus somewhat resembling the plasma membrane one. The core subunit, the potassium permanent subunit, is called MITOK. The original name of the gene is CCDC51. If you look in mice, you have one unique isoform, while in humans we have two different isoforms. There are two splice variants. We still have to understand the specific role of each of these isoforms. And then there is an accessory subunit that now we call MITOSUR, because it really resembles to the SUR subunit of the plasma membrane KATP channel. It belongs to the same family. It is an ABC containing protein. It is called ABCB8. It was known as ABCB8. Now we renamed this subunit as MITOSUR. Even in this case, you have a unique isoform in mice, while 4 different isoforms, again, they are splice variant, are present in humans. Thus meaning that most likely the MITOK ATP activity is somewhat more complex, must be more complex and more tightly regulated in humans compared to other organism models. In any case, despite all these variants, they share the overall organization, that they contain transmembrane domain, the same transmembrane domain, and the same ATP binding domain. And what we demonstrated that in situ, you can have an interaction of this channel. Here you can see on the left an immunoprecipitation. On the right, you can see a native gel showing that there is a complex at around 500 kilodalton that can react both with MITOK and MITOSUR antibodies. Thus, most likely the stoichiometry is for MITOK subunit and for MITOSUR subunit, again somewhat resembling the plasma membrane KATP channel. In any case, the demonstration that this complex can form a channel, of course, relies on heterologous systems. So we reconstituted these proteins in planar lipid bilayer. And as you can see here, if you reconstitute only MITOK in planar lipid bilayer, you can see nice channel activity that is somewhat potassium specific. And however, if you put ATP, you can see that MITOK alone lacks ATP sensitivity. However, when you reconstitute both MITOK and MITOSUR together in the plasma, in the planar lipid bilayer, now you can see the standard pharmacology of the MITOK ATP. So you can see that the channel is inhibited by millimolar concentration of ATP. And if you on the same planar lipid bilayer, you put diazoxide, the opener, you can see the channel activity back, as you can see in this figure. And as to blockers, you can see here that both glibenclamide, so sulfonylureas, and 5-HT can block the channel. So the molecular complex made by MITOK and MITOSUR can recapitulate the main pharmacological features of what we know, what it is intended to be the MITOK ATP. And in cells, you can see that the MITOK protein is strictly located in the inner mitochondrial membrane. And this is something specific because all other potassium channels reported to be present in mitochondria are usually intended to be splice variant, or are channels that are both present in the plasma membrane and in the inner mitochondrial membrane, while this protein is localized strictly in the inner mitochondrial membrane and not to any other intracellular membranes. You can see here the protein is enriched in mytoplasm, and it is an integral membrane protein because it can't be extracted by carbon extraction. But most importantly, we also wonder what is the physiological, or what is the activity, in situ activity of this channel. So we overexpress this channel in normal HeLa cells, okay? And what we realized that the overexpression of this channel, of the MITOK subunit alone, causes a dramatic mitochondrial phenotype. As you can see here, MITOK overexpressing cells has fragmented mitochondria. If you look by electron microscopy, you can see that mitochondrial completely swell. That is in line, of course, with increased potassium entry. And if you look to membrane potential, of course, as you can imagine, these mitochondria are completely dysfunctional. They have any more mitochondrial membrane potential. Also mitochondrial calcium uptake, that of course depends on the existence of any of mitochondrial membrane potential, is of course dramatically reduced in MITOK expressing cells. And of course, this is in line with the fact that MITOK ATP is the potassium permeant subunit that can't be inhibited by ATP, as we demonstrated in the planar lipid bilayer system. So, however, the idea is that if we put back the regulatory and inhibitory subunit, we can have a normally gated MITOK ATP channel. So we should recover the phenotype triggered by the overexpression of MITOK alone. And indeed, here, for example, you see a mitochondrial membrane potential. You can see that MITOK overexpression alone causes loss of mitochondrial membrane potential, but if you put the normal physiological subunit, you can see that you can recover normal mitochondrial membrane potential, and this is true also for mitochondrial calcium handling. So this demonstrates that also in vivo, also in cells at least, MITOSUR can inhibit the potassium permeant subunit and reconstitute a normal function, the normal function of MITOK ATP. As to the physiological functions, to the role, the role of the endogenous protein, what we did was to develop and knock out cells for MITOK, for the potassium permeant subunit, and usually these cells has not a very evident phenotype, but if you look carefully to mitochondrial function, you can see that there is a widening of the crystal, and this is in line with an impairment of the regulation of the matrix volume as expected from a potassium channel. If you look to membrane potential, you can see that membrane potential is there, but there are this transient depolarization. So there is an instability of mitochondrial membrane potential in MITOK knockout cells, so cells lacking MITOK ATP activity. And if you look to oxidative performance, again, you can see that the respiration of this cell is somewhat impaired. Again, in a case, this phenotype is specific because if you put the channel back, you can see that you can almost recover the normal respiration. However, then we move to the mouse model. So we developed a global knockout model. As I told you, cells has no any major phenotype, and also the MITOK, the global MITOK constitutive MITOK knockout mice shows almost no overt phenotype, because you see normal, that mice are born at normal Mendelian ratio. They, at least until 4 months of age, they are pretty normal, with the same weight and everything. In any case, as I told you at the beginning of my talk, there are several evidence that MITOK ATP, as other mitochondrial ion channels, plays a critical role in ischemia reperfusion injury. Indeed, several ion channels, for example, including, for instance, the mitochondrial calcium uniporter or the mitochondrial permeability transition pore, has been clearly demonstrated to be important in mediating the reperfusion injury. And there are pharmacological evidence that also MITOK ATP is involved in this process. So we tested this. So first we used a pharmacological preconditioning model. So this is a model, an ex vivo model, when you take the heart, you perfuse the heart either with vehicle or with a MITOK ATP opener, such as diazoxide, and then you measure the heart injury after ischemia and and reperfusion. As you can see here, as reported by other, diazoxide is an effective treatment to prevent reperfusion injury. However, this pharmacological preconditioning triggered by diazoxide is almost completely lost in the case of MITOK knockout hearts. And along the same line, also ischemic preconditioning is a way to protect the heart. In this protocol, you have to perform some brief ischemic episode, then you do the long ischemia and then the reperfusion. And in normal condition, you can see that this is a protective treatment. And again, also in this, with this experimental setup, you can see that the loss of MITOK dampens this protective effect triggered by ischemic preconditioning, at least in part. Thus meaning that this channel is indeed important for ischemic precondition. So the take-home message is that we have identified this molecular complex located in the inner mitochondrial membrane. This is a potassium channel that is sensitive to ATP concentration. It is normally closed because physiological ATP concentration can close the channel. And what we know that potassium homeostasis control the regulation of swelling and contraction of the matrix, thus meaning that mitochondrial potassium can regulate the volume. This in turn leads to the remodeling of the crystal and the tuning of oxidative phosphorylation. So thanks everyone. Of course, there are several outstanding questions, so we still have to identify other maybe component of this MITOK ATP. We have to, of course, investigate whether ATP is the only modulator of this channel. And of course, there is the most, I think, important question is that we still are missing what is the normal housekeeping role of this function. And of course, we have to also figure out the molecular mechanisms that are activating during cardioprotection mediated by this channel. So I have to thank, of course, several colleagues that participate in this study. For instance, Fabio for all the work in the heart, and Ildi with all the electrophysiological experiment, of course, Sarino Rizzuto for the support on everything else. And of course, all our lab, and I hope you have some questions for me. And I'm open, of course, to address all your questions.
So thank you so much, Diego. Very, very, very interesting talk. In fact, that was mirrored by Rajesh, who's in, and everybody's reminded, please type your questions in questions and answers box, by Rajesh, who said, great talk. Do we know if any patient-associated mutations exist in MITOSUR ATP-sensitive domain or elsewhere?
Okay, thank you. So this is an interesting question. As far as I know, of course, I look to available databases, available variants, and up to now, I still couldn't find any variant that is associated with some kind of pathology. Of course, I think this is somewhat normal because, of course, now that we have the challenge, maybe people will start looking at that. But up to now, I'm not aware of any mutation associated to pathology.
Thank you. We also have a comment from one of the world's experts in KATP channel, Colin Nichols, who says, fascinating. I agree. So we also have a question here from Enrique Balderas. I hope I say the name correctly. The question is, the MITO-BK, whoop, this thing is jumping around. The MITO-BK-CA knockout, this is the NCNMA1 knockout hearts, do not show any cardioprotection compared to wild type hearts. And he references Singh et al. In 2013. And the question is, these investigators haven't reported a compensatory role for MITOK ATP channel in a MITO-BK channel knockout heart. What is your thought about this?
So this is an interesting question. Thanks for asking this. So this, of course, the MITO-BK channels is one example. In any case, there are other examples than when you somewhat interfere with one mitochondrial potassium channels, you somewhat have some kind of compensation, or at least you see some other potassium conductance that are increased. So we are still have to, in principle, this could happen, this could occur also in our model. Of course, we do see a phenotype, of course, at least during ischemia reperfusion in the context of ischemia reperfusion injury. And of course, maybe we lack any evident phenotype because maybe in some other channel could compensate for that. In any case, we are planning some patch clamp experiment to see whether in our MITOK knockout mitochondria, some other conductants are hyperactivated. Again, this is true for many other potassium channels, so somewhat I'm expecting that other conductants for potassium will be upregulated in our knockout model.
Okay, thank you so much. So there are a few other questions as well. I'm not sure how far we're going to get. Noriko, you have to stop us when we go over time, please. Not only did Colin say very interesting, but he also, let me just go back here. He understands that you don't have structure yet, but we do know that the detailed structure info of the SUR1 CARE 6.2 channel. What is the primary comparison between MITOK ATP and MITO-SUR and SUR plus with CARE 6.2?
Oh, excellent question. So, so the overall molecular organization looks between the mitochondrial and plasma membrane looks similar. However, the point is that while the source immunity is somewhat similar, so they belong to the same family. So again, both the plasma membrane and mitochondrial source immunity are ABC containing proteins. In the plasma membrane, you have ABC-C8 and ABC-C9, while in the mitochondria, you have ABC-B8. So they look pretty the same. I'm pretty sure that they share a significant structural homology. However, the potassium permanence immunity is completely different. So the MITOK and the KIR subunit are very far one from each other. And as a matter of fact, the MITOK channel has no classical selectivity, potassium selectivity filter, the KYG. And along this line, I have to say that at least in planar lipid bilayer, the specificity against potassium is not, so the channel is not that specific for potassium. So in general, I think that the source unit and accordingly the pharmacology that is based on the source unit is pretty similar, but the potassium permeant subunit I would expect is very different, and we know nothing about that. So we have to do structural studies and also mutagenesis studies in order to understand what are the key residues that enables channel permeation.
I think that your answer you just gave also answers the question of Kevin Foskett, who wanted to know a little bit about the transmembrane prediction of MITO-SUR. Let's go to the question of Bimel Desai. He asks if the ATP, when the ATP levels drop, the activation of MITOK should decrease the driving force for calcium influx, but that seems counterintuitive because calcium influx drives mitochondrial metabolism. Any reconciliation ideas?
So thanks for this question. So first of all, according to our topological studies, the ATP binding domain of the MITOSUR subunit should be located in the intermembranous space. So this channel should sense the cytosolic ATP concentration. And so, of course, and we know that mitochondrial calcium stimulates intramitochondrial metabolism, and so at first it increases the matrix ATP concentration. In any case, I would also comment that on one way, we know that ATP acts as an inhibitor of the channel. However, I think that maybe other endogenous molecules could activate. So what I do believe is that closure by ATP is a sort of mechanism to keep the channel closed most of the time. I'm not totally sure that the drop of ATP is the endogenous stimulus that opens this channel. Because if you look to ATP sensitivity, still at the 200 micromolar is sufficient to close the channel. And there are no physiological conditions where you have less than 200 or even half a millimolar ATP concentration inside the cell. So I do believe that ATP is an inhibitor of the channel that is necessary to keep the channel closed and block any futile cycle. But I do believe that the opening is mediated by something else. But of course, this is just gut feeling.
So Diego, there are a bunch of other questions. I really urge you to look at the Q&A box and please answer as many of those as you can. We are out of time, as you can see that the slide came up here, and I'm going to pass the floor now to Stefan, who's going to introduce the next speaker.
Great, thank you, William. Our next speaker is Reza Sharif Naeini from McGill University in Montreal in Canada, and he will talk about a new channel that senses pain. And many of you will know that there is a number of ion channels that are involved in pain sensation in response to thermal stimulation and also to mechanical stimulation. There are voltage-gated sodium channels, there are potassium channels, there's TRPV1 and others. But Dr. Naeini identified a new channel that's involved in the mechanical pain sensation. And I thank you for joining us and presenting your data today. It's all yours. Dr. Sharif?
Yes, just going to share my screen. Okay, very good. So thank you very much. Thank you for the introduction, Dr. Fesque. I want to also take this opportunity to thank Drs. Fesque and Koedzi for organizing this event, as well as Ms. Noriko Shimizu for the coordination and Ms. Caitlin Simpson of the Biophysical Society for facilitating this event. So in case I run out of time, I just quickly want to highlight the trainees and the colleagues who have helped really make this work possible. So the two main important trainees for this work were Lou Beaulieu-Laroche, who's at MIT right now, and Dr. Marine Christin, who's still at McGill with us right now. And then great colleagues, Dr. Kate Poole in Australia, Dr. Ricard Blanc at the University of Montreal, Dr. Emmanuel Bourrinet in Montpellier in France, and Dr. Ribeiro da Silva at McGill University here. And of course, the funding agencies, the Rita Allen Foundation, the Canadian Institutes of Health Research, the National Science and Engineering Research Council of Canada, and the McGill Pain Center. So thanks for bearing with me. It was just important for me that these get the credit that they deserve. So my lab is interested in the mechanisms that underlie our senses of touch and pain, from the peripheral sensors all the way to the networks in the spinal cord that process these inputs. At the periphery, we know that the detection of touch and pain inputs relies on a process called mechanotransduction, where mechanical inputs are converted into electrical signals. And because of the rapidity of this process, it is understood that it must rely on the activation of mechanosensitive ion channels. These are channels that will convert mechanical forces into electrical signals for the nervous system to be able to process. And we know from years of either single channel recordings or wholesale recordings that the channels that are responsible for the sense of touch and pain are likely different in nature. And we know that based on their single channel conductances. So the channels that have been recorded from touch sensitive primary neurons typically have a single channel conductance between 20 and 30 picosiemens, whereas those that have been recorded from pain sensing neurons have much lower single channel conductance between 10 to 13 picosiemens. And then when we record them at the wholesale level, these experiments are wholesale recordings in which a blunt probe is approached near the cell and does mechanical indentations on the cell body. And what we see is that there's a rapid adaptation to this mechanical stimulation when the recording is made from touch-sensitive neurons, and the activation threshold is actually quite low. So low threshold rapid adapting currents are typically what are recorded from touch-sensitive neurons. When we record from pain-sensitive neurons at the wholesale level, what we see is that the kinetics of the current are very different. So they display a slow adapting behavior and typically have a high threshold for activation. And that's important because, you know, for touch sensing, you want these neurons to rapidly adapt so that your central nervous system is not constantly bombarded with information that it doesn't necessarily need. Like when you put on a shirt, for example, or when you put on your watch, you don't need your nervous system to constantly be reminded that there is a watch on your wrist. But the sensation of pain is essential for survival. So if you are in a potentially injurious situation, it's important that this information never desensitizes. So you need your nervous system to be constantly reminded that there is a potential injurious stimulus being applied. Now, as to the molecular identity of these sensors, we now know from compelling evidence, mostly from the group of Arden Paraputian at the Scripps Research Institute, that the gene responsible for the touch-sensitive neurons is piezo2. And they have a long list of experiments and manuscripts in which they have demonstrated this, including in vivo experiments in which they used an inducible knockout of piezo2 and were able to show that the mice responses to mechanically applied stimuli to their feet was very different at low intensity stimuli, but as you increase the intensity of that stimulus, then you would lose that difference. But the identity of the channels responsible for pain sensing remained mostly elusive. So over 10 years ago, a study was published in which there was a proteomic screen of membrane proteins involved in mechanotransduction. And several candidate mechanosensors were identified in this screen, including one called TMEM120A. Now, we renamed this TACAN for the Farsi word movement, because we think it may have some role to play in detecting mechanical stimuli. So TACAN is highly conserved among species. Here you see a sequence alignment of human, mouse, and zebrafish sequences. And we have stretches of over 20 amino acids that don't change by a single residue. And when we look at its conservation across different species, we can go far back to Drosophila, C. elegans, Arabidopsis, Thaliana even, and they all have an ortholog of TACAN being expressed, suggesting that it must have been important to be conserved throughout evolution like that. And when we look in mice at the tissue expression of TACAN, we see that it is expressed in several tissues and with high expression in the heart, kidney, and colon, as well as the dorsal root ganglia. So the dorsal root ganglia, or DRGs, are where we find the cell bodies of the sensory neurons involved in the detection of touch and pain. So we generated an antibody, which we affinity purified against TACAN, and we tested it in knockout animals as well to verify its specificity. And when we looked at its, so what you're looking at right now are cross sections of dorsal root ganglia in which you find all the different types of sensory neurons. So there will be proprioceptors, which can be identified with an antibody against parvalbumin, and they can also be touch sensitive neurons or pain sensitive neurons. And when we look at the different patterns of colocalization, we see that there is a high co-expression of TACAN in pain sensing neurons. So in the sensory neurons, in the dorsal root ganglia, there is a high expression of TACAN in pain sensing neurons. So this was, you know, for a channel to be involving pain, you needed to be expressed in the pain sensing neurons. So this was our first line of evidence for this. Now, the most important question is, if we think it is a potential mechanosensitive candidate, then we should be able to express it in heterologous systems. And when we mechanically stimulate these cells, we should see an increase in mechanically evoked currents. And there are many different ways to assess mechanosensitivity in vitro. Two of the most common ones that are used are either the cell-attached recordings, which you can see on the left. And in this case, it's a cell-attached recording. The membrane at the tip of the electrode is left intact. And then we apply brief pulses of negative pressure through the recording electrode, and this is going to cause a distension in the membrane. If there are mechanosensitive channels present in that patch of membrane, they're going to open, and we will be able to record the current flowing through them. The other approach is a whole cell approach. So we do a whole cell recording and bring a blunt probe to poke the cells and record whole cell currents. And both have their advantages and disadvantages. So when we express TACAN in CHO cells, we see that MOG transfected cells have their background level of endogenous mechanosensitive channels, but when they express TACAN, then we have a significant increase in mechanically evoked currents. And this is what you can see in the middle panel where the average data is expressed. When we express TACAN in HEC cells, once again, we see a significant increase in mechanically evoked currents. And the group of Ardem Paraputian had determined that the main mechanosensitive channel in HEC 293 cells is piezo1, and they generated a piezo1 knockout cell line. So we took those cells and introduced TACAN in them, and we're able to see that, once again, presence of TACAN significantly increases mechanically evoked currents. Now, these are all in cell-attached recordings. The problem with going to whole cell recordings is that a lot of these expression cell, heterologous cell lines are very flat. So it's difficult to do these mechanical indentations. If you have a low threshold activated channel, like piezo, for example, then you can see these small membrane deflections causing large inward currents. But if you have a high threshold activated channel, then odds are that you would be breaking the cell membrane before you can see the first channels opening. So it was difficult for us to see any wholesale current with this approach. But we turned to our colleague, Kate Poole, who had developed this assay in her lab, and also when she was a postdoc in Gary Lewin's lab, where she cultures cells on these pillars. And then she can do a whole cell recording from coming from above the cell, and then take a stimulation probe and gently deflect these pillars and record whole cell evoked currents. And she demonstrated that by recording from native cells, such as touch-sensitive neurons in green, in the green symbols, or nociceptors, pain-sensing neurons in purple. And what she saw was that deflections as little as 10 nanometers were enough to activate mechanically evoked currents in touch-sensitive neurons. And then if you increase the intensity of that deflection, you start getting whole cell currents in the pain-sensing neurons. So we sent her our constructs. She transfected it in the piezo1 knockout HEC cells, and she was able to determine that when you increase the intensity of that deflection, now you suddenly start seeing these large inward currents, which are slow adapting and have a high threshold for activation. So you can see the white circles are piezo1 knockout HEC cells transfected with a mock plasmid. So in these ones, we can go as far as 1 micron of deflection of those pillars, and we never see any currents. But if they have TACAN expressed in them, now we go to 100 or 200 nanometers of deflection, and we can see these large inward currents. So keep in mind, in native cells, in the nociceptors, the endogenous mechanosensitive channels has a high threshold for activation and a slow adaptation kinetics. And now we take that channel and we take that candidate and put it in a heterologous systems, and it reproduces 2 of those important characters. So high threshold for activation, as well as slow adaptation kinetics. To verify that TACAN forms a channel, we turned to our colleague Ricard Blanc, and what we did was to purify TACAN and reconstitute it in synthetic lipids. When we did, we saw that, so these are planar bilayer recordings. When we inserted TACAN containing proteoliposomes on that bilayer, we suddenly see these macroscopic currents that displayed an ohmic behavior. And if we take these currents and apply known blockers of mechanosensitive channels, such as GSM-TX4, we immediately block these channel openings. Okay, so it indicates that TACAN is expressed in nociceptors. It can increase mechanically evoked currents in cell lines. And if we purify it and put it in synthetic lipids, it does form an ion channel that can be blocked by blockers of mechanosensitive channels. The next test is to see in cells that endogenously express TACAN, like the nociceptors, what happens if we downregulate the expression of TACAN. So we took native nociceptors from mice, and we transfected them with siRNA molecules that were conjugated to Alexa fluorophores, so that we could visually identify which nociceptors had taken the siRNAs and record from them. So what you're seeing on the left is illustrative traces from nociceptors that have been transfected with a non-targeting siRNA on the left or an siRNA targeting TACAN on the right. And what you can see is that once we increase the intensity of that mechanical stimulus on the left, we start seeing these channel openings. But in the presence of TACAN, we have a significant decrease in mechanically evoked currents in these nociceptors. Now, it's possible that TACAN is also expressed in touch-sensitive neurons, and maybe it's not really specific to the nociceptors. And one way to look at that is to do wholesale recordings from a large population of sensory neurons. So like I showed you in the beginning, touch-sensitive neurons will display a rapid adapting current, whereas the pain-sensitive neurons will typically display a slow adapting current. So if you record from a large population of neurons indiscriminate, without focusing on nociceptors, then some of those neurons will display a rapid adapting current, and others will display a slow adapting current. Okay? And there's a certain proportion of these. If you look in this bar graph in the middle. So when we record in from sensory neurons transfected with the non-targeting siRNA, we have about 30% of the neurons that display a slow adapting kinetic, displayed in the red section, whereas about 30 or so have a fast, rapid adapting current. But when we knock down TACAN expression, what we see is that the slow adapting currents, the cells displaying slow adapting currents are almost completely gone, suggesting that TACAN seems to be contributing to these slow adapting currents in DRG neurons. Removing TACAN embryonically is lethal, so we had to devise a way to selectively remove it from nociceptors. And to do that, we used a mouse line in which the nociceptors express an inducible Cre recombinase. And what we did was to put these mice on a wire mesh platform and then stimulate the plantar surface of their paws with small filaments of varying force. So these are called von Frey filaments, and they come in a large variety, going from very small force being applied, as 8 milligrams, something that you or I would never be able to feel, all the way to 300 grams of pressure. And each filament is applied for 5 seconds on this plantar surface and 5 times. Then if there's no response, we move to a stronger filament and a stronger filament until we start seeing some behavioral responses. And as we increase the intensity of these filaments, the mouse will either not move its foot or start gently stretching its digits. So it means that it detected it, but it's not bothering the animal. If we increase a little bit more, what we see is a reflexive withdrawal. So the mouse will quickly lift its foot and put it back down again. And these kind of behaviors, we can also see it on mice that have been transected at their spinal cord level. So we think it's largely a spinal reflex, okay? That's why we call it a reflexive withdrawal. But if we increase the intensity even more, now not only the mouse will lift its foot up, but it's gonna shake it or lick it. And these are behaviors that are typically associated with pain behaviors, okay? So if you look at the graph in panel A, we're only looking at the displays of reflexive withdrawal. And if you look at the black symbols, what happens is that as you increase the intensity of the force being applied, you get more and more of these reflexive withdrawal. But as the intensity increases even more, we see less of them because now they're replaced by pain-like responses. Okay, we call them nociceptive responses. Now, this is what happens in a control animal. But if we remove TACAN from these mice, they continue to display just a reflex withdrawal, despite the fact that the intensity of the stimulus is increased, which suggests that they may not feel the pain as much, or they continue to feel this as if it was a touch type of response, okay? And if we specifically focus just on the pain-like responses in panel B that you can see, that's where you can see clearly that there's a significant decrease in nociceptive responses. So the mice have a defect in sensing pain-like stimuli. This is shown in the bar graph in the bottom of that slide. So here we just looked at the responses to a 1 gram pressure being applied to the foot of the animal. And we record the percentage of the time where the mouse will display pain-like behaviors. Okay, so if we just take the flux animals without the Cre recombinase, then adding tamoxifen doesn't change the distribution of the pain-like responses. These are the red sections that you can see. But if we took an animal, a TACAN fluxed animal that is crossed to a Cre-inducible driver line, we see that after injection of tamoxifen, there is a significant reduction in the proportion of pain-like responses, which means that decreasing the expression of TACAN in nociceptors impairs the ability of the mice to detect painful stimulus. Now we have gone on to look at this from different approaches. If we take a wild-type animal and inject it with an AAV vector that carries an shRNA against TACAN, and we do this injection in the sciatic nerve bundle, so these are the nerve fibers that innervate the foot of the animal, and we only do it on one side of the animal, leaving the other side as control. We see that the injected side will have, again, loss of detection of painful mechanical stimuli, whereas the touch-like responses are unaffected. And if you can see in the lower panel, if we take our flux animal, and again, in the sciatic nerve bundle, we inject an AAV that expresses the Cre recombinase under neuronal specific promoter, synapsin. Once again, we can replicate the same observation, which is that the animals seem to specifically lose their ability to detect mechanical pain. So I haven't been keeping track of time. I hope I didn't go too fast. So in summary, and I kind of went over some of the other findings that we have, but in summary, TACAN appears to be a novel ion channel. When we looked at its sequence, we were able to find any known domains, which makes our job of further characterizing this channel even more difficult. So it is expressed in nociceptors, and expressing it in cell lines will increase the cell's responses to mechanical stimuli. If we remove it from cells that endogenously express it, we see that, like in nociceptors, we see a significant reduction in intrinsic mechanosensitivity, and removing TACAN in adult mice will reduce their responses to painful mechanical stimuli without affecting their sensitivity to heat, to touch or heat. I didn't show you heat responses, but heat responses are unaffected in these mice. So with that, we come to the conclusion that TACAN is an ion channel that is essential for sensing mechanical pain. But there are a lot of questions that are still remain to be answered. So we're still working on determining whether this channel is intrinsically mechanosensitive or not. And if it is, we have some structure function studies to do to determine what are the molecular determinants of mechanosensitivity in this channel. We are searching for partners of TACAN that could be responsible for its mechanosensitivity, and a list of other questions which we are answering. So with that, I thank you for your attention, and I'm happy to answer questions.
Great, thank you, Reza, for this fantastic talk. There are a couple of questions, probably others coming in. So let me start with one from Sahil, who is asking, what is the habituation response of these channels in, or in other words, do repeated stimuli evoke unabated responses?
So we have done this with intervals of about 10 seconds, but if we, our pulse protocol in the cell attached recordings is for about 1 second, and the channel can respond as soon as within 5 milliseconds of the beginning of the stimulus. In some instances, it will stay open for the entire duration, for the entire 1 second. In others, it can shut on and off, on and off. Overall, the probability of opening will increase with stimulus intensity, but it does not adapt to the stimulus being applied, if that was the question.
Yeah, exactly. So maybe that is related to the last question by Amanda Klein. What is the role of TACAN in chronic pain signaling?
Right. So, you know, in that first study that we published this year, we didn't have time to go over all the different pain symptoms, and this is what we're doing now. There are different types of chronic pains, some that are due to changes in the spinal cord circuitry, whereby touch inputs are going to activate pain circuits. And then there are other chronic pain symptoms in which the pain sensing neurons, which normally have a high threshold for activation, will become sensitized and their activation threshold decreases. So now a gentle touch, for example, is going to activate pain sensors. And we've shown in a previous study on osteoarthritis pain that during some of these pathological conditions, it's the activation threshold of the mechanosensitive channels that is decreased. And because they're expressed in pain sensing neurons, that means that low intensity stimulus is going to activate the pain sensing neurons. So our hypothesis, and we have a few pain models running right now, is that in pain models in which the activation threshold of nociceptors is decreased, we would see a contribution of TACAN to this pathology.
Yes, and that is maybe again related to a question by Gerd. I guess that's Gerd Bulting, asking, is there an impact of inflammation on TACAN activation and expression? Right. I guess that would play a role for these chronic pain, inflammation-related syndromes.
Yes, absolutely. So in collaboration with a group in Montpellier right now, we are examining all the potential phosphorylation sites in the cytosolic domain of TACAN. We think that to get a link between inflammation and a potential decrease in the activation threshold of TACAN, in the gating properties of TACAN, intracellular cascade are involved. So either phosphorylation of TACAN or modulating its interaction with the cytoskeleton. So these are two things that we are investigating. But an important point is that we don't think that during pathological conditions, you will see a significant increase in the expression level of TACAN because, and this could be one of the reasons why it has eluded genomic screens that have been done over the years, because people have a tendency to look at the top 3, top 5 upregulated candidates. But depending on where's the membrane resting potential of these neurons and how much single channel current you get from opening just an individual channel, you don't need an enormous amount of channels to be activated to get neurons to fire action potential. Something like 50 channels is enough. So in this case, we don't need to increase the number of the channels. We just need to modulate their gating properties. And that's enough to trigger chronic pain conditions.
There's another question from Joseph Farley about, is TACAN also expressed in the brain in the expected places, or is it just in peripheral nociceptors?
No, it is expressed in the brain as well. We haven't looked thoroughly at its expression level, but there is a group, I believe, in France that is currently looking at that, and the preliminary data indicates that there are expression.
Okay, great. Sure, Noriko, you have to stop me when we are running out of time. Are there mutations in human patients that are associated with TACAN?
So we are collaborating with the genetics lab here at McGill to identify those, but we haven't found any yet. So I don't know what that means, to be honest with you. And there are a lot of questions. Is it that it's so important that any loss of function mutation would be lethal? We don't know. It's not my specialty either to really comb through this, but we're hoping to find them. We're hoping to find perhaps gain of function mutations in patients that have chronic pain and see whether those have increased sensitivity to pain. So it's ongoing, but they're all very good questions. Yeah.
Maybe I can ask one last biophysical question that relates to the activation mechanism of the channel, and that's related to the to a comparison with piezo channels that that interact with the deforming membrane. But it looks like that TACAN has actually a fairly short N and C terminus. Do you know how it senses the deformation of the membrane?
So there could be two ways. Two classical ways have been identified. One is via interaction with cytoskeletal elements or elements of the extracellular matrix. The other one is via lateral tension in the membrane. And the idea that had been proposed is that during membrane distension, you could have a slight thinning of the bilayer, and this would expose hydrophobic regions of those transmembrane domains, forcing the channels to change their orientation in order to re-embed the hydrophobic residues in that membrane. So this is the case for mechanosensitive channels of bacteria. These are, the way they gate is all the subunits will turn as the shutter of a camera. And in doing so, they can re-embed their exposed hydrophobic amino acids within the membrane again, and this causes an increase in the pore size. So we don't know exactly. One of the experiments that we're doing is to identify the pore of the channel, and we're doing that via these the SCAM analysis, these cysteine mutagenesis approach. And we're hoping to get more information on some of these critical functions in the very near future.
Yeah. Great. I think we have to move on, but there's more questions in the Q&A box of in Zoom. So please have a look, and maybe you can answer some of them by responding directly. Thank you again for the great talk, and I hand it over back to William for the next speaker.
Thank you, Stefan. So it is my pleasure to introduce Dr. Zhaozhu Qiu from Johns Hopkins University. Talking of genetic screens, genomic screens, they've indeed done a genomic screen and identified one of the TMEM proteins as a novel protein-activated chloride channel that's involved in cellular responses to acidosis. And this was recently published in Science. We're looking forward to what you have to tell us.
Thank you for the nice introduction, and organizers. It's a great opportunity, and the talks are very exciting. So we are interested in chloride channels. You might wonder why we should care about chloride channels. Actually, chloride is only major negative charged ions in our body. And in the last 50 years, people typically focus on cation channels, sodium, potassium, calcium channels. For good reasons, their underlying action potential and signal transduction. However, chloride is also very important in many functions. And it might be very surprising to you that many chloride channel hasn't been cloned, meaning that you can do electrophysiology and measure the current density, but you don't know what genes are responsible for them. So my lab has been, in the last 10 years, using function genomics to discover new chloride channels, developing imaging assays, and combined with really powerful genetic approaches such as siRNA, CRISPR. And we discovered two new families of chloride channels. The first is SWEL1 or LRRC8 family, encoding the volume regulated anion channel. And the second is a PAC channel, which is acid activated. So I briefly touch upon those two channels. So all the mammalian cells in our body has this remarkable ability to regulate volumes. And when the cell is in a hypertonic situation, the cell will swell by osmosis, and then all the cells can recover from that swelling. And it was thought this was done by a process called volume regulatory decrease. And basically the cell will express salt, particularly potassium and chloride. And the major component of this pathway is a channel called VRAC. It can release this chloride, and it's called anion channels. They are not really selective. So a lot of chloride channels are not very selective because they don't have to. The chloride is the main anion inside of the body. So they can also release osmoles such as taurine inside of cell. And when this osmoles released by osmosis, the volume will come back. However, the molecular identity of this VRAC has been a long mystery. And I will not go into details. Basically, that we performed ionized, whole genome ionized screen. I will tell you the assay in a minute. And if you treat cell with swelling and you have this assay, this is a control scrambled siRNA, and this is a blocker. And this is a secondary screen we did after genomic screen, and one gene popped out as the best hit possible. It's a little known gene called leucine rich repeats containing family 8A. We call it SWEL1 because it's activated by cell swelling. And this was simultaneously discovered by both our group and Thomas Jentsch's group in Germany. And it's always good to have reach the same conclusion. It quickly became clear that this channel is important cell volume, regulation, but cells don't typically experience hypertonic. So the many stimuli can activate the channel. And one of the key characteristics of this channel is that they can not only conduct chloride, but also many large organic osmoles. In particular, we have been focusing glutamate in astrocytes. It's important for releasing of glutamate to regulate neuronal excitability and synapse transmission. We also had a collaborative study. Recently, we found that these channels can also conduct the cGAMP, where it's regulating the innate immunity. And I think the next speaker, Chris, will talk about this in detail. He also has a very beautiful story. So for the remaining part of the talk, I will just switch gear to talk about another channel which we identified recently. It has to do with acid. So acid is widespread in physiology. Many vesicles are acidic, and many places, some tissues can get acidic during disease. There's many acid-activated receptors was found previously, but there's also a acid-activated chloride channel is also kind of very common. So the way this was discovered is that if you do a whole cell patch, you have a patch pipette, and you perfuse acid in this case, and you get this classic very large outward current at plus 100 millivolt. And you can appreciate that this channel is activated by perfusion of acid extracellularly, so most likely the acid sensing is outside. I will come back to later this point. And this channel has been recorded in many cells, a large variety of cells, suggesting that it might have some important functions in cell biology. However, it also suggests that this channel might have something to do with acid-induced cell death and tissue damage, as illustrated here. That in the ischemic brain, locally it will become acidic, and the pack is activated. The chloride will go into the neuron and cause the neuron swelling and cell death. However, to validate these findings, we need to identify the molecular identity. And in order to do that, we turned into this YP quenching assay. It has been successfully used to screening drugs for cystic fibrosis, for example. It has a very simple principle where this mutant version of YP, when it's binding to anion, particularly iodide, the fluorescence will be dimmer. And we reason that if we make a stable cell line, and we know that PAC is expressed in those cells, and if you add acid and also put iodide in a solution, as I mentioned, that the chloride channels are not selective, so they can also let iodide into the cell, so then the fluorescence will be quenched. And that's exactly what we found. So here is the normalized fluorescent traces where when you're adding acid plus the iodide, the acid opens the channel, the iodide quenches the fluorescence. So you get this nice dose-dependent curve. So we're ready to go to do a fishing expedition. We're very lucky to get a library where, because we try to search for an ion channel by definition, it has at least 2 transmembrane domains. It actually, there's a finite number of these genes in the human genome, about 2,700 of them. So we're lucky to get a library where we were targeting each gene with 4 siRNAs. And this is typical fluorescent traces where if you zoom in most cases, the PAC is expressed, so you will get quenching because the channel open, iodide get in, so you get quenching. But occasionally, you also get less quenching, and those presumably could be its channel, could be a channel regulator, and those would be a target you will be focusing on. And we're very excited that we're getting very good at this assay that just did the primary screen. We found a very exciting novel membrane protein. So this is about 3,000 genes. So each one has 4 siRNAs. So it's about 12,000 individual siRNAs. And typically, most of hits, you only have 1 siRNA working. But this is the only one which has multiple. In this case, 3 out of 4 siRNAs are highly active. So it's a very generic gene. There's no papers on them, called TMEM206, and that's just the best case. So we call it the PAC for proton-activated chloride channel. So it has 2 transmembrane domains and looks like a channel. And before we get in business, we first want to make sure that in the recording situation, the current is really gone. So that's my postdoc, Junhua, who did the recording. Again, you patch a cell, perfuse acid, you see this classic PAC current. And in the meantime, you also see this inward current. This is mediated by ASIC, which is the acid-activated cation channel. And it can be blocked by aminoride. So both, this is the current traces for the voltage steps for the PAC current. It's very typical. So we made a knockout cells, and both in 293 cells and HeLa cells, in both cases that the outward PAC current are completely gone. And importantly, the inward ASIC channel is still present. So it's a specific inactivation of the PAC. And this is the traces also show the channel is gone. The next question is that now you identify gene which is necessary, it's required for the channel activity. Is this sufficient? So we made a PAC human cDNA and expressed in a knockout cell. So this is a vector, there's no current, but in the knockout cell, we'll get a huge amount of current, much bigger than endogenous current. And they looks very similar to the endogenous current. In addition, the EC50 is also similar. And it's interesting, this channel can be modulated by temperature. So at 37 degrees, the EC50 shift to the left, make it more physiological in the pH 6 range. We also did more characterization that look at the ion selectivity. So these channels are a little bit more selective for iodide than chloride, and it's the case for both the cDNA and the Y type, and they also can be blocked by the typical blocker, non-specific chloride channel blockers. And this is the evolutionarily conserved protein all the way to the vertebrates, so to the zebrafish. So we look at the zebrafish PAC and see whether it also encodes the acid-sensitive channel. And indeed it did. And it not only responding to the acid, it also exhibits some interesting features. So remember, this is a fish cDNA expressing human cells, but it encodes a slightly different property in that the human PAC, when the voltage changes, the facilitation is very quick, and the fish PAC slightly slower. And when you go back to the voltage, the fish has a little bit larger tail current, meaning the open and close a little bit slower compared to the human channel. And they also responding to pH, but has a different Hale coefficient, and they also has slightly different ion selectivity. And this would suggest that PAC would likely to be a pore-forming subunit. Although I will not go into too much details, where my student James did extensive mutagenesis and demonstrating that mutating PAC can have effects in ion selectivity, which is a poor property. In this time of age with the advance of cryo-EM, that we thought that the definitive answer really is that can we see the channel? And we're lucky to form the very great collaboration with the Wei Lu's group at the Van Ander Institute in Michigan. And they are great structural biologists, especially spearheaded by postdoc Zhen Ren. And so he purified the protein and put in nanodisk and determined the near anatomic resolution of the structure. First at pH 8, which is a resting state at 3.6 Angstrom. I was just kind of blown away by the structures. You remember, this is just after months or a couple of years after we identified the sequence, and now you see a channel. It's just blew me away. So it's a beautiful structure, like a bouquet. It has two transmembrane domains and a head. It looks, it's a trimer, and it is, thank goodness it's channel. It actually very much look many other two transmembrane domain channels, particularly A6 channels. There are some differences, but overall structure is similar. I would emphasize they also don't have sequence homology, so there might be some evolutionary convergence happening here. So then went ahead to also solve the structure at acidic pH 4, where it will generate a proton bound state. I will draw attention that we found a large conformation change in the TM1 domain and also the linker region between TM1 and extracellular domain. So I'll play this video. So histidine is very good for sensing pH because the pK is around 6, so it will be protonated when the pH drops. And we found a big movement from a resting position to the acidic pocket about 5 angstrom away. So again, this is pH 8. This is the top view from the channel. And this is a histidine 298. In the resting state, interacting with this residue, and then it flipped over with interacting with the acidic pocket, which form this, and this is the basis of the swing of the TM1 domain. And it has this remarkable domain swapping where the TM1 initially interacting with the TM2 in the same subunit, but now it's interacting with the adjacent TM2. So there is a small iris movement in the extracellular domain. It's just very, very beautiful and satisfying to see. Great. So we made mutations in some of the acidic pocket I was illustrated to you, and all of them has shifted the EC50 to the left, meaning they are more sensitive. However, none of them individual mutations will give you fully activated current. So we think that there may be multiple pH sensors as very common for many other acid-sensitive channels as well. So we look at ion conduction pathways. This is pH 8. It's a resting closed state, and by previous studies, we demonstrate TM2 is the main pore lining domain, which is the case. And in pH 4, although there's some opening up in some restrictions, however, overall it's still a non-conducting state. We speculate that this might be a desensitized or a pre-open state. And we noticed that there is a positive charge lies in 319, which lies in the perfect position, which could regulate ion selectivity. And James went on to make a mutation to change the lysine to glutamate, where did the charge reversal. And interesting, this, if you focus on the black line, this is the Y-type current, and this is the mutant current. Now it becomes a inward current, and this inward current is not mediated by chloride, but by sodium. So when you change the extracellular solution to 15 millimole sodium chloride, for chloride channels, it will shift to the right, and for sodium channels, shift to the left. And if you calculate the selectivity, it completely changes the selectivity from a chloride selective to a sodium selective. So we believe that this lysine determines the anion selectivity for PAC. So the last question is what is really the role of the PAC? You know, there's many diseases associated with acidosis and where it could play a role. Obviously, one of the place people typically think about is stroke, where you have acidosis, chloride load cells, and cause cell swelling death. And indeed, the PAC is widely expressed, but also highly expressed in the brain. So we look at that. First, we tried in the cell line 293 cells. We treat the cells with acid. It caused necrotic cell death. However, in the knockouts, both cases, this death is protected. And if you overexpress PAC, you sensitize the cells to more cell death. So then we made a PAC knockout mice, and these mice had some behavior phenotypes, but overall they are viable. So we made a isolate primary neurons. Again, that when we treat the neurons with low pH, the cells where there's a percentage of cells will die. And the knockout, we found there's significant protection from that acid-induced neuronal cell death. So the next step is really try to look at this in an animal model where you can basically do a surgery and put a filament into the mouse brain, and it will block the mid-cerebral artery for a number of times. And this will cause the mid-brain necrotic injury. And typically that this core region is very difficult to salvage, but in the surrounding regions, you might have a way to rescue those cells. And indeed that Jana, my postdoc, who did this blindly, where she looked at both wild type and knockout, and she found that although in the core regions, there's very little difference, but in the surrounding parenchyma, called parenchyma regions, there's significant improvement of protection for the ischemic induced brain injury. And those mice also do well in neurological scores as well. So I was just going to summary that chloride channels has many functions: the regular membrane potential, the regulated membrane potential, and fluid secretion, and also cell-cell communication. And the PAC is a new acid-sensitive channel, and we think it plays a key role in acidosis. And swell channels release and uptake diverse single molecules, and we look at glutamate, cGAMP, and also ATP. They're likely to regulate many signaling pathways. And swell and PAC are potential drug targets for disease associated with pathologic cell swelling and acidosis. And we really appreciate the powerful approaches of functional genomics, which you can use RNAi or CRISPR. It's very effective decoding their identity. And there's still many chloride channels hasn't been identified, in particular as alluded to that in the intracellular organelles, for example. People who involved, and really special appreciation for Wei Lu and Du group in Michigan, where they solved the structure for PAC, and it's great collaboration, which is a merge of structure and a function. And we are funded by NIH and several foundations. We're also, now with those new channels, we're really trying to figure out what they do and how they function. So we are actually recruiting postdocs as well. Thank you very much.
Well, thank you very much, Dr. Qiu. It's really very, very nice work. I agree with you that the new structural information is just beautiful, and it is very informative. Thank you for including those data as well. There are some questions. I'm going to start with the question of Peter Lauf, who asked whether you can expand on the mechanism of temperature dependence of PAC activation and what the time constant is of relaxation if you return the cell to a low temperature.
Yeah, we haven't looked at it carefully. So, it's something that, we would want to look at maybe in the future. It's very interesting because if you recall that room temperature, the cells, the channel are less sensitive, to the pH drop. So that's, it's a good question. We should look at that in the future.
Thank you. So the next question you might have started to address already, but Sahil wants to know that the pore doesn't look very wide, being only 1 angstrom at the narrowest. And whether you have any thoughts on iodide conduction through this extremely narrow crevice.
Yeah, so this basically the channel. We think it's not in a full open state. So this is not an open structure, and we know that, for example, we did SCAM analysis with MTSCS. It definitely can access lower part of the pore. So the pore in the open state, it will be much bigger than this.
Thank you. Kevin Foskett would like to know whether PAC is expressed in epithelial cells or on the plasma membrane, subcellular localization, apical localization, basolateral. Any thoughts on that?
Yes, that's a good question. You know, I think the PAC is expressed in many cell types, you know, including epithelial cells. But we are, unfortunately, antibody is not very good. So we're making a, you know, TAC mice try to see, addressing these very interesting questions. Thanks.
Okay, thank you. Fabio Echeverria would like to, well, first congratulate you on beautiful work, but he asks whether you have checked what tissues express PAC channels, and I think you've partially answered that question, but which parts of the brain in particular would express PAC?
Yeah, so PAC we look at is expressed in neurons and expressed in microglia and a little bit low levels in astrocytes. So it's you know, it's really widely expressed.
Thank you. Joseph Farley has a question whether you can comment on the roles of PAC versus ASICs in MCAO ischemia.
Mm-hmm. Yeah, I mean, you know, so the background, the A6 channel was also previously implicated in ischemia because they were load cell, they were be activated and conduct calcium. So I think that based on our data, you know, both of the channel might be both participating, I would say, because you know, knockout either one of them have some partial protection. So it will be interesting maybe if you can block both of them, you might have some synergistic effect.
Okay. And then we have a last question from the audience, it seems, from Dr. Jiang, who wants to know whether PAC alone, when expressed in bilayers, was it able to form a channel?
Yeah, that's something that my students would love to do. That's from all the evidence we have, it looks like you don't need anything else.
Okay.
To you know, to see the channel particularly strongly. I think that, you know, if you express a fish version of the channel in a human cells, you get a different property. So that again suggested maybe this is essential pore forming itself.
Excellent. There is a last question by Geert, who wants to know whether there is a any relation with the connexin 3 hemi channels because they contribute to the spreading of cell death in ischemia reperfusion.
Can you say it again? Sorry.
So the connexin 43 hemi channels.
Oh, hemi channels. Yeah.
Yeah. Is there any relationship with the hemi channels of connexin 43 because they do contribute to the spreading of cell death in ischemia reperfusion?
In terms of genetic.
That is not specified. So please just.
Yeah, I mean, so for the you know, sequence itself, there's no link to any known channels, even A6 channel. You know, even the structure are very similar, but there's no sequence, not too much of sequence homology. Okay. But it will be interesting to see. You know, sometimes the channels can be localized together to facilitate the process. So it's still possible, I would say.
I'm going to sneak in one final last question here, and that is whether you have any insights in specific pharmacology for these channels.
Mm-hmm. Yeah, so that's one of the issue for the chloride channel research is that there's lack of very specific blockers. There's two reasons. One of the reasons is that you don't know the molecular identity, so you couldn't get them. Mm-hmm. And second is that it's just historically, you know, people, I guess they don't look, you know, deep enough. For example, toxins, right? You know, we typically see them to targeting sodium channels or potassium channels. So I think that would be a very interesting area to explore, you know, look into pharmacology of them. And one of the issue being plagued the field is that, you know, people will treat a blocker, but then the, you know, they will just block all kinds of chloride channels. So that's it's something that impeding the progress of the field.
Right. Well, I think we're out of time, so I really would like to thank Dr. Qiu for a wonderful talk, and I'm going to hand the chair or the floor back to Dr. Feske.
Thank you.
Yeah, thank you, William and Dr. Qiu. That was great, and we're actually continuing. This will be a very smooth transition to our last speaker, who is actually a trainee, Chris Ritchie from Stanford University, where he is a graduate student in the lab of Lingjun Li, and I think he will tell us about a story that's related to the channel that you actually discovered with, I think, Adam Putaputian, and this is the LRRC8 A to E channel family. And this is interesting because it was published this year that these channels play a role for the transport of the immune modulator cGAMP in macrophages. Now, the lab of Lingjun Li published last year that another transporter, SLC19A1, can also transport cGAMP. And I think we'll hear about that today, and probably also about another heterodimer of LRR family, LRRC8 family members in immune transmitter transport, which is actually a fascinating topic in innate immune modulation. And so I'm really looking forward to this talk. Chris, welcome, and thank you for sharing your data.
Thank you for that introduction and giving me the opportunity to speak at this event. Yeah, so our bodies are just constantly encountering threats to our health, and these threats can come from the outside, such as in the case of viruses, as well as from within, such as in the case of cancer. And thankfully, these threats are often associated with specific danger signals that can be detected by our innate immune system in order to eliminate these threats by eliciting immunity. Now, there are a lot of different danger signals that our innate immune system can detect, but one specific danger signal that is associated with a broad array of threats is the presence of DNA in the cytosol. So, for example, double-stranded viruses, when they infect cells, can often release DNA into the cytosol. And also, cancer cells frequently will have DNA within their cytoplasm as a result of chromosome missegregation. So when DNA is present in the cytosol, it's detected by the enzyme cGAS, which upon binding to DNA becomes active and synthesizes the small molecule 2,3-cGAMP, pictured here. cGAMP will go on to bind to its receptor STING, which results in kinase activation and phosphorylation of the transcription factor IRF3, which ultimately results in transcription of interferons and activation of the immune system in order to deal with the threat. Now, the interesting thing that has come out of the Li lab recently is that not only is cGAMP able to activate STING within the cell that is producing cGAMP, but Jackie Carosa and Volker Bonnert recently showed that cGAMP can be secreted from cGAMP producing cells and into the extracellular space, and then subsequently imported into nearby responder cells that are then able to activate their STING pathway to activate immunity, thus propagating the immune activation signal. And one important feature of this paracrine signaling is that often cancer cells will knock out immune activation as a result of STING pathway activation. And so transmission of cGAMP to nearby cells is sometimes the only way that immunity is activated as a result of cytosolic DNA presence. Unfortunately, when we found this out, we actually had no idea how cGAMP was transmitted between cells. And specifically, we're interested in trying to determine how cGAMP is imported into responder cells. So Anthony Cordova in the lab a while back now discovered that cultured cell line U937 cells will die in response to extracellular cGAMP. And this seemed like a good observation to exploit in order to try to identify the cGAMP importer. So to exploit this observation, Anthony Cordova teamed up with Galen Hess in Mike Bassick's lab to design a U937 CRISPR library, and subsequently treated this library with multiple rounds of cGAMP to enrich for cells that were infected with guides that knocked out parts of the STING pathway. And presumably some of these guides would be targeting the unknown transporter of cGAMP in these cells. So here is a figure showing the results of the screen. So the CASA score is just a measure of the enrichment of different guides at the end of the screen. And the most enriched guides are known members of the STING pathway, including STING itself, really validating the approach of this screen. And another interesting hit was this transporter called SLC19A1, which we thought might be a potential cGAMP importer given its place on the screen. And so to begin to try to figure out if this hit was a cGAMP importer, we knocked it out of U937 cells and tried to assess whether or not it affected extracellular cGAMP import. And as a proxy for measuring extracellular cGAMP import, we would look downstream at STING signaling events. So in this case, we're looking at IR3 phosphorylation, which is downstream both of STING activation and of cGAMP import. And so if 19A1 is a cGAMP importer, we would expect that knockout would reduce IR3 phosphorylation. And indeed, this is what we saw. So knockout of SLC19A1 and U937 cells decreased the phosphor-IR3 response to extracellular cGAMP by about 75%. However, this doesn't necessarily mean that 19A1 is a cGAMP importer, because it could be affecting some process of this IR3 phosphorylation downstream of cGAMP import. And so to really get at the question of whether or not SLC19A1 is involved with cGAMP signaling at the level of cGAMP import, we performed this experiment where we bypassed the need for import by just simply electroporating cGAMP into cells and then measuring IR3 phosphorylation. And the idea is that if 19A1 is indeed an importer and only affecting signaling at the level of import, that knockout shouldn't have any effect when cGAMP is introduced into cells by electroporation. And what we saw was that when cGAMP is electroporated into cells, SLC19A1 knockout has no effect on cGAMP signaling, indicating that SLC19A1 is affecting extracellular cGAMP signaling at the level of cGAMP import, which is strong evidence for it being a cGAMP importer. One thing that I haven't mentioned yet is that SLC19A1 has previously been characterized very well as a folic acid importer, and it's also known to import the anti-cancer drug methotrexate, which can act as a competitive inhibitor of other SLC19A1 substrates, and it's known to be inhibited non-competitively by the compound sulfasalazine. And so we decided to use these tools to really get at, to really increase the evidence we had that SLC19A1 is indeed a cGAMP importer by asking whether or not these inhibitors inhibited extracellular cGAMP signaling. And what we saw, again, is that both compounds, sulfasalazine and methotrexate, resulted in strong reduction in phosphor-IR3 response to extracellular cGAMP. And importantly, this decrease was dependent on the presence of SLC19A1. So when SLC19A1 is knocked out, we don't see this decrease in phosphor-IR3 levels. And so all these results together indicated to us that SLC19A1 was the cGAMP importer in U937 cells and was, in fact, the first identified cGAMP importer. And a group also independently identified SLC19A1 as a cGAMP importer around the same time that we did, which really gave us confidence that we were correct in assigning its role as a cGAMP importer. However, one of the interesting things that we observed was that when we knocked out SLC19A1 in U937 cells, there still was residual phosphor-IR3 signaling. There still was presumably cGAMP getting into cells despite us knocking out the cGAMP importer, which suggested to us that multiple cGAMP import mechanisms must exist. And so I'm not going to actually go into detail too much about this, but I will say that Lauren Leahy in the lab performed a CRISPR screen in U937 cells that had SLC19A1 knocked out in order to identify this residual cGAMP import activity. And what she found was that SWEL1, also known as LRC8A, complexed with its paralog LRC8C or LRC8E, was able to function as a cGAMP importer. And as Zhaozhu mentioned, this was another cGAMP importer that was independently identified by another group. So moving away from looking at cGAMP import in U937 cells, we wanted to try to determine how cGAMP is imported into other cell types, because one of the observations we had made in our studies of SLC19A1 was that different cell types seem to use different cGAMP import mechanisms. And there were specific cell types that we were very interested in trying to identify the import mechanisms of. I don't have a chance to show the data behind this cartoon, but Anthony Cordova and Volker Bonner in the lab were able to perform in vivo experiments to identify which cell types are actually responding to tumor-derived extracellular cGAMP in vivo. And they identified 5 cell types that are able to respond to tumor-derived cGAMP. And those are classical monocytes, M1 macrophages, endothelial cells, NK cells, and T cells. And so while we know SLC19A1 is the importer of U937 cells, we were unsure of what the importer was in these other cell types. Lauren Leahy was able to demonstrate that LRRC8A C complexes were responsible for cGAMP import in endothelial cells, but that still leaves us with 4 cell types to try to characterize. And a cell type that I was particularly interested in was classical monocytes, because they're easy to isolate from donor blood. And so the first question I decided to really get at was whether or not SLC19A1 was the possible cGAMP importer in this cell type. And to do this, I just saw whether or not the SLC19A1 inhibitors, sulfasalazine and methotrexate, had an effect on extracellular cGAMP signaling in these cells. And got some interesting results. So while one of the inhibitors, sulfasalazine, inhibited extracellular cGAMP signaling, the other SLC19A1 inhibitor actually didn't appear to have any effect on extracellular cGAMP signaling in CD42 monocytes. And so if SLC19A1 was the importer in the cell type, we'd expect both inhibitors to be able to inhibit extracellular cGAMP signaling. But it seems that something else is going on in these cells as far as cGAMP transport is concerned. So one thing that I thought was interesting was that there are actually multiple splice isoforms of SLC19A1. And one splice isoform, SLC19A1-202, which is different in its last exon, is known to not be able to import methotrexate. So I was wondering if perhaps, if both isoforms are inhibited by SSZ, but only SLC19A1-202 is not inhibited by methotrexate, perhaps SLC19A1-202, this alternative splice isoform, is the importer in CD14 monocytes. So to test this, I decided to overexpress this isoform in U937 cells and see if it affects extracellular cGAMP signaling. And to my disappointment, this isoform actually had no effect on extracellular cGAMP signaling in this when it was overexpressed, indicating that it's not a cGAMP importer. I still thought this was an interesting result to follow up on, and I decided to look outside of SLC19A1 at other proteins. So the fact that CD14 monocytes are inhibited by sulfasalazine, but not methotrexate, it indicates that there must be some transporter in these cells that is targeted by sulfasalazine that isn't in U937 cells, since U937 cells are only inhibited by sulfasalazine when 19A1 is present. And so I decided to look at some expression data that previously exists that compares transcript levels of plasma membrane transporters in CD14 cells versus U937 cells, with the idea of looking for transporters that were enriched in CD14 cells. And one transporter really jumped out, which is SLC46A2. It was highly expressed in CD14 positive monocytes, but wasn't expressed at all in U937 cells. And furthermore, it's closely related to a known target of sulfasalazine, SLC46A1. So I was wondering if this protein could potentially be the importer in CD14 monocytes. So just like I did with the alternative transcript of SLC19A1, I decided to overexpress SLC46A2 in U937 cells and see if it affects their ability to respond to extracellular cGAMP. And I was very encouraged by this, and I saw a very, very strong increase in IRF3 phosphorylation when SLC46A2 is overexpressed. Furthermore, when I saw whether or not SLC46A2 had any effect on electroporated cGAMP, found that SLC46A2 overexpression does not affect the response to electroporated cGAMP, indicating that 46A2 is affecting cGAMP signaling at the level of cGAMP import, indicating that it's a cGAMP transporter. Now, to bring it back to CD14 monocytes and trying to get at the question of whether or not it is the dominant import mechanism in these cells, I wanted to see if 46A2 was indeed inhibited by sulfasalazine. And here are the results for that. So again, U937 cells, when 19A1 is knocked out, are not inhibited by sulfasalazine. And when 46A2 is overexpressed, there's normally a very large increase in signal that is reduced back to baseline when sulfasalazine is present, indicating that sulfasalazine inhibits 46A2. However, there could be other targets of sulfasalazine in CD14 monocytes. And so to really try to nail that 46A2 was the target of sulfasalazine in CD14 monocytes and is the dominant import mechanism in these cells, I knocked out SLC46A2 with Cas9 RNPs and then measured how much SSE was able to inhibit cGAMP signaling in these cells. So I wasn't able to get complete knockout in these primary cells, but I got multiple samples with a range from 40 to 50% 46A2 knockout. And in these samples, I saw that the ability of SSE to inhibit cGAMP signaling was decreased by roughly half, indicating that 46A2 is the target of sulfasalazine in CD14 cells, and that 46A2 is likely the cGAMP importer in this cell type. So going back to this cartoon of responder cells to tumor-derived cGAMP, we were able to identify 46A2 as the dominant cGAMP importer in classical monocytes. And I don't have time to show these data, but we're also able to show that it's the dominant import mechanism in M1 macrophages that are derived from classical monocytes. However, there still are some unknown responder or some unknown transport mechanisms for responder cells that we're interested in following up on and trying to really complete this picture of how cGAMP is differentially transported by different responder cell types. So yeah, I just want to acknowledge everyone in the Lee Lab for being along with me in this story, especially Anthony Cordova, who has been my partner for pretty much all the work that I've shown today. Also want to thank the Basic Lab for helping us set up CRISPR screens that were essential in identifying SLC19A1 and LRRC8 as cGAMP importers. And with that, I'll take any questions that exist.
Great, thank you, Chris, for the very nice talk. We have a couple of questions that are currently coming in. One question is about the relationship between the SLCs that you described and the LRRs, C8 and C proteins. Could it be that SLC19A1 might be involved in activating the transporter instead of being the transporter per se, and potentially by activating the LRR proteins?
Yeah, that's a very interesting question. So I didn't mention this, but LRRC8A channels are activated by a wide variety of stimuli, and it could be that sting activation does lead to LRRC8 channel opening. That's something that we haven't had a chance to look at yet, but it's a very interesting idea that I think we want to look into.
But is there data that that if you express the SLC19A1 or SLC46A2 that they can form pores? For instance, if you express them in lipid bilayers, can they transport cGAMP under those conditions?
We haven't been able to express these proteins in lipid bilayers. So, yeah.
Okay. A related question is, are there cGAMP binding pockets in these candidate proteins?
So, I mean, we don't really have structural data on SLC19A1 or SLC46A2, so I can't really answer whether or not there are any surfaces that might bind to cGAMP in those proteins. Lauren has done some beautiful mutagenesis experiments to show that mutating certain residues in the pore of LRRC8 are able to prevent cGAMP transport. So presumably there is a cGAMP binding pocket in the LRRC8 ion channel.
I see. Then there are two questions about the cGAMP response by T cells. Thomas Comolo is asking, what is the T cells response to cGAMP? And maybe a related question is, any thoughts which might be the cGAMP importers in NK cells and in T cells? Is SLC46A2 expressed in these cells?
Okay. Yeah, so well, I'll start with the first question. So T cells are known to die after prolonged exposure to extracellular cGAMP. And to kind of follow up on that, that's one of the reasons why we think it's interesting that different cell types might have different transporters with different affinities. So it's possible that the T cell transporter has a much lower affinity for cGAMP, so that it's only importing cGAMP when there's very, very high extracellular cGAMP concentrations around. And it could be that cGAMP-induced T cell death is a mechanism to prevent hyperactivation of the immune system. Mm-hmm. We don't think that SLC46A2 is the T cell or NK cell transporter just based off of expression data. So SLC46A2 expression is actually very limited to just a few cell types in humans. So it's highly expressed in monocytes and then monocyte-derived cells such as macrophages and dendritic cells, but not really expressed in many other cell types.
Maybe I can ask a question about the selectivity of these SLCs and also the LRR channels for 2,3 cGAMP, right? These are produced by eukaryotic cells, tumors, as you mentioned, but they are also similar molecules that are produced by prokaryotic organisms like bacteria, 3,3 cGAMP or C-di AMP and GMP. Did you test the selectivity of these different transporters for these molecules?
Yeah, we've looked at a panel of cyclic dinucleotides for all of our transporters. And we found that, well, I'll start with LRRC8 channels because they're probably the easiest to summarize that LRRC8 channels appear to be very selective to 2,3 linked cyclic dinucleotides, but at least the human homologs don't appear to be responsible for uptake of 3,3 linked cyclic dinucleotides. That is cyclic dinucleotides produced by prokaryotes. SLC19A1 and SLC46A2 are both able to import both 2,3 and 3,3 linked cyclic dinucleotides with differing selectivities for the various cyclic dinucleotides.
Okay. So Kevin Foskett is commenting, that's more a comment than a question, I think, saying not your work, but LRRC8 channels have big pores and can release big molecules, so one wouldn't expect them to be constitutively active in endothelial cells. I think that's a comment on the Leahy paper that you said is accepted, I think. So any comment on that?
Yeah, I mean, we aren't really, yeah, so we were able to show that hypoosmotic treatment of cells, when LRRC8 is activated, endothelial cells appear to use LRRC8 as a cGAMP transporter, and we've activated them with hypoosmolaric shock. And it's possible that in vivo there are other stimuli that activate LRR in order to enable it to import cGAMP, but that's something we haven't fully characterized yet.
I see. Then there's one question by Joseph Farley saying, I'm curious as to how cells infected by positive strand viruses like SARS-CoV-2 and others, I guess, herpes viruses, in which subgenomic DNAs are being produced at high levels and the cytosol manage to evade C-gas sting signaling. I mean, this may not be directly related to the transport mechanism, but any idea how these viruses can escape the response?
So SARS-CoV-2 is, I believe, an RNA virus. I don't think...
Yeah, if you talk about herpes viruses, then maybe, right?
Yeah. I think that some ways that herpes viruses or other viruses that have some DNA component as their genome are able to evade detection by cGAMP is simply by protecting their genome from the enzyme cGAS. So, for example, lentiviruses, I believe, will have capsids that form around their genome, and these capsids protect the genome from exposure to the cytosol and will deliver the genome into the nucleus. So I think just simply by preventing exposure of these genomes to the cytosol, viruses can prevent activation of this pathway. And an alternative mechanism that some viruses use is to actually knock out components of the sting pathway. So, for example, I think herpes virus, at least some herpes viruses, have proteins that will abrogate sting signaling.
Mm-hmm. Okay, great. Then I have one last, there are a couple of more questions, but I will just close with a more biophysical question by Bimal Desai, who is asking, since the inward transport of cGAMP is not favored by the membrane potential, which is negative, obviously, and cGAMP is a negatively charged molecule, are the high extracellular concentrations actually ever achieved in vivo, high extracellular concentrations that are required for import of a negatively charged molecule? How much, and is this to what degree are these mechanisms in vitro responses? And I guess the question is how much evidence is there that these mechanisms, these cGAMP import mechanisms, play a role in vivo as well?
Yeah, so we haven't been able to directly measure extracellular cGAMP concentration in vivo. However, based off of our in vitro data, we do believe that it can get to concentrations at which it is able to be imported by responder cells. And along those lines, we have found that when we neutralize extracellular cGAMP by injecting basically a tool that will bind and sequester extracellular cGAMP in tumor, we do see an effect on the immune response, strongly suggesting that extracellular cGAMP is able to get to high enough concentrations to be imported by responder cells.
Okay, great. Thank you very much for answering these questions so thoroughly. There are a couple of more questions. You may want to look at them and then respond to them. I think we'll wrap up the symposium now. We are right on time, and I thank all the speakers for staying on time and for the really fascinating talks. I think we covered a really nice spectrum of unusual channels and transport mechanisms, and I found this very stimulating. I hope the attendees also found this interesting. And I thank all the attendees for asking great questions and engaging with the speakers. So before we finish, I just want to take 2 or 3 minutes to with a couple of closing remarks. And one is to point your attention to an initiative or consortium that's sponsored by the NIH, and that's called the IDG, or Illuminating the Druggable Genome Initiative, or consortium. This is sponsored by the NIH Common Fund. And the goal of this initiative is to study human proteins for which publicly available information or active research is really is lacking in order to identify new drug targets in the end. And there are currently three protein classes under study, and one of them are ion channels. And if you look at their website, you will see that there's about 100 or so ion channels that are considered dark and included on this IDG list of proteins to be studied. And so the title of our symposium was not entirely accidental. We were inspired by this IDG initiative, and that's why we called our symposium Shedding Light on Dark Ion Channels. And I would encourage you to go to this website of the IDG consortium and look at their resources and the different components of this program. They have a data resource generating center, a knowledge management center. They have bioinformatics tools. There's a great database called the Pharos database, and I included the URL of this database here, and that will help to assess how much information is available about certain proteins, and some of them are, and some of these understudied proteins. I think this is a great resource. There's also, at least until recently, funding mechanisms or several funding mechanisms associated with this initiative, including our 3 grants. I would also like to point out a conference that we are organizing. Next slide, please. That we are organizing next year. This is the 74th annual meeting of the Society of General Physiologists, and this has a very specific topic, and that's about ion channels and transporters in the immune system. And I think, especially in light of the last talk by Chris Ritchie, some of you may be interested in this conference. We wanted to hold this conference this year in Woods Hole in September, but because of COVID-19, we just had an online symposium a month ago, and we postponed the real meeting as a 4-day conference to next year. So we have 4 days of talks by really outstanding scientists who work on ion channels in the immune system. We'll have poster sessions for postdocs and students. We'll have career panels and so forth. So if you're interested in this, please look at the website. And if you're interested in this topic, and this is kind of a bit of a self-serving advertisement, the next slide, at NYU, we're building a new program which is called Ion Channels in Immunity, and there are currently 2 open positions, one for the director of the Ion Laboratory, an electrophysiology core facility. If some of you may be interested in that, let me know. There's also an open faculty position to study ion channels and transporters in immunity. And again, if you're interested, please contact me. With that, I thank my colleague William Coetzee, Noriko Shimizu, Caitlin from the Biophysical Society, and all of the speakers and attendees for coming to this meeting and participating. And I hope you found this inspiring, and hope to see you all again soon. Bye-bye.
Thank you. Bye.
Bye. Take care, everybody.